Good morning, everyone. Welcome to the NIH press briefing on therapeutics discovery. I'm John Berklow, Communications Director at the NIH. I will moderate the Q&A following the speakers and the presentations. Just a couple of simple ground rules. One, since this is a press briefing, I ask only credentialed reporters ask questions. And number two, please silence your cell phones. Now it gives me great pleasure to introduce our first speaker, Secretary of Health and Human Services, Kathleen Sebelius. Since taking office in 2009, almost exactly 3 years ago to the day, Secretary Sebelius has led ambitious efforts to improve America's health. Under her leadership, HHS is committed to innovation, from promoting public-private collaboration to finding novel ways to speed life-saving medicines to the market, both of which are topics of today's event. Madam Secretary.
Well, good morning, everybody, and thank you, John, for that introduction. I'm delighted to have a chance to be here with some of our distinguished partners. You will hear from Dr. Francis Collins in a little bit, a great leader at the National Institutes of Health. At the table also are Donald Frail, who is the Vice President of Science, New Opportunities, and Innovative Medicines Unit at AstraZeneca. Dr. Jan Lundberg, Senior Vice President of Science and Technology at Eli Lilly, and the President of Lilly Research Laboratories, Rod McKenzie, Senior Vice President, Head of Pharma Therapeutics Research and Development at Pfizer, and Michael Manganello, who is a patient advocate. And you're going to hear Kathy Hudson also is joining us at the table. When we look back over recent history, what we know is that biomedical research is responsible for some of our greatest progress, from the discovery of penicillin to the development of effective therapies for cancer. Diseases that had once been a death sentence have been eradicated or cured. Conditions that once had been disabling are now manageable. Now, these achievements haven't only saved lives and improved the health of millions, but they've sparked enormous economic growth and created countless new jobs. At the beginning of a new century, we can see even bigger opportunities ahead. Last week at the White House, I joined Cabinet Fellows to introduce our nation's ever first bioeconomy blueprint. This administration's commitment to strengthening bioscience research as a major driver of American innovation and economic growth, and keeping America on the forefront of the search for new cures and treatments is fundamental to achieving that vision. And yet the road from research lab to an approved and marketable drug is a long one. There are many detours and obstacles along the way. It can take many years and hundreds of millions of dollars to bring one new drug from discovery to the marketplace. And most new projects fail in early stages. If we want to find cures for the 21st century, we need a chart to a clear path forward. And that's why this administration has undertaken a major effort to identify and remove roadblocks at every step of the way in the development process. Last December, under the leadership of Dr. Collins, we took a big step forward with the creation of NIH's National Center for Advancing Translational Science, or NCATS. The center's mission is to develop the tools and approaches we need to help everyone searching for new cures to move forward at a faster pace. One of the center's earliest priorities has been to take advantage of the many pharmaceutical industry compounds that were originally developed and tested for one particular purpose, but ultimately not approved. Now, what we know is that many of the compounds may have other potential uses. We just need to do the research to identify them. The drug AZT, for instance, was originally tested against cancer and failed. Only later, it was discovered to be a very effective treatment for HIV, the first medicine we found to work against the virus. And Michael, who's with us today, will talk about what that discovery has meant for him. In the search for new cures and treatments, these compounds offer a big advantage over those still being developed in the lab. They've already cleared many of the clinical and regulatory hurdles. We know that they've cleared the toxicity hurdle, allowing researchers to identify new therapies more quickly and allowing companies to bring them to market more efficiently. And yet many compounds, way too many compounds, haven't been reevaluated. Or if they've been reevaluated, the research is typically conducted through traditional partnerships of very limited size and scope. But today, we're pleased to announce that that is changing. I'm proud to announce a new collaborative program that will allow far more researchers to study pharmaceutical industry compounds and pursue vital new treatments for patients. Now, the goal is simple: to see whether we can teach old drugs new tricks. And to get there, we're taking an innovative approach, crowdsourcing these compounds to our brightest minds and most inventive companies. Under the initiative we're launching today, any researcher with a promising scientific idea can apply for a new grant to test compounds from our partners against a variety of diseases and conditions. Through the program, the National Institutes of Health intend to provide at least $20 million in fiscal year 2013 to support these new grants. And for their part, our partners in the drug companies will provide researchers with access to compounds and all the related data. Now, this new partnership is an investment, not only in our researchers, but in our nation. Because when American scientists have the tools and resources to pursue the next great discoveries, we all benefit. It makes our nation stronger, healthier, and more competitive. And I'd like to now introduce a critical leader in that partnership, somebody who has had his eye on this possibility for a period of time, the great leader of the National Institutes of Health, Dr. Francis Collins. Dr. Collins.
Thank you very much, Madam Secretary. It's an honor to have you with us here this morning for this exciting announcement. We have come a long way in understanding the causes of disease, especially in the last few years. As a result of the genomic revolution, we now know the molecular basis of about 4,500 diseases. That's an amazing thing to be able to say. But moving such discoveries into treatments has been far more difficult. Of those 4,500, only about 250 of those diseases have treatments. Why is this so? Well, the triple frustration of long timelines, steep costs, and high failure rates have bedeviled the translational pathway. It currently takes an average of $2 billion and 14 years to move a discovery from the lab to the clinic. And of thousands of compounds that enter the drug development pipeline, only a very few will ultimately make it to approval. We should, in fact, collectively, we must do better. By working together as a community, patients, public sector, private sector, I believe we can. As Secretary Sebelius noted, the public-private partnership we're announcing today is the first signature initiative by NIH's new National Center for Advancing Translational Sciences, or NCATS. In this effort, we will seek to discover new therapeutic uses for existing molecules through an innovative alliance with Pfizer, AstraZeneca, and Eli Lilly. In this pilot phase, the program will match U.S. researchers with dozens of these companies' compounds to explore how they might be put to new uses in treating patients. And NIH is happy to serve as the matchmaker. The compounds that the companies are providing to this program are the result of their significant investment in research and development. Tens of millions of dollars and years of work have already been invested in each compound. They've already cleared safety testing in humans, but did not prove effective for the specific disease or condition for which they were developed, or they were not pursued for business reasons. But thanks to the initiative being announced today, researchers from U.S. universities, biotech companies, and nonprofits can apply for NIH funding and access to these compounds in order to test their effectiveness in treating other diseases or conditions. There are numerous compelling historical examples of why this strategy will work. The Secretary already mentioned the story of AZT. and we'll hear more about that later from Michael. But there are others as well. Another good example is raloxifene, a drug developed for osteoporosis that has now been shown to be as effective as tamoxifen in preventing invasive breast cancer. And for its part, tamoxifen is now showing effectiveness for the treatment of bipolar disorder. There are numerous other examples of compounds that were approved for one disease but are now being used to treat others. But we need to generate more of these success stories in a more systematic manner. As the Secretary said, we need to crowdsource the solutions. Because the compounds being brought forward in this new collaboration have already been tested in humans, detailed information is already in hand regarding dosing, pharmacology, and toxicity. By building on this preclinical and clinical information, new applications for potential therapeutics can be identified and validated far more quickly and efficiently than if one had to start from scratch. So here's the plan. In the first year of this program, NCATS will provide up to $20 million in grants to researchers across the nation. Any researcher with a promising scientific idea can apply for one of these grants to conduct preclinical and clinical feasibility studies to test compounds from our partners for their effectiveness against a variety of diseases and conditions. For their part, the companies will provide researchers with access to the compounds and all related data. A key aspect of this partnership is the use of innovative template agreements. By providing such standard template agreements between the company and biomedical research partners, this program will significantly reduce the amount of time it takes for each of the partners to negotiate the terms. Traditionally, those negotiations have been required on a case-by-case basis for each project, which can take significant effort and time for both parties and can even result in failure to launch the project at all. Too often, it has taken longer to negotiate the terms of the agreement than to actually do the research. These template agreements will be a standard that can make this kind of relationship go much more smoothly. As part of the template agreement, pharmaceutical firms will retain ownership of their compounds. The research partners will own any intellectual property they develop through the research project and be able to publish the results of their work. I want to point out that this alliance was conceived with the help of the FDA-NIH Leadership Council, chaired by Peggy Hamburg and myself, which asked a working group in 2011 to identify challenges and opportunities in drug repositioning. The work of that group has helped shape our thinking and led to this initiative. Such visionary thinking is imperative if we want to take bold action to speed up drug development. To that end, I want to extend my sincere personal thanks to these three pharmaceutical firms for thinking outside of the box and providing unprecedented access to their compounds. We at NIH hope this is just the beginning of many more great things to come, that the pioneering spirit shown by these companies will inspire others to join us in this important effort. That invitation is wide open. So to summarize, this program is not just a win-win, it's a win-win-win. It's a win for investigators who get access to valuable compounds for new uses. It's a win for companies whose compounds have a chance for new applications and new life. And most importantly, it's a win for patients whose hopes for discoveries of new treatments are significantly advanced by this partnership. Now, I'd like to introduce you to my Deputy Director of Science Outreach and Policy and the Acting Deputy Director of NCATS, Dr. Kathy Hudson. Kathy, you played a leading role in shaping NCATS and this first NCATS initiative. Can you just say a word about how this program exemplifies what NCATS is all about?
Francis, and I want to add my thanks to the folks at the table and their teams, some of whom are in the room, who have worked really hard to make today a reality, so thanks to all of you. The mission of NCATS, which is only 4 months old, is to catalyze the development of cool new tools that will speed the delivery of new medicines to the patients who need them. And I think this initiative is an incredibly cool new tool. We hope that we'll be able to get other pharmaceutical companies to join us, and that this will be an effective means of matching promising compounds to the very best, brightest ideas in our biomedical crowd. The other thing that we're experimenting with is how well do these deals work? We pre-negotiated these templates. We have some variables in this template, so we're doing an experiment on how to be able to accelerate these kinds of multi-stakeholder collaborations.
Thanks, Kathy. And at 10 o'clock this morning, the request for information, as well as the template agreements that I mentioned, have gone live. So all of you who are interested in seeing the details may do so beginning this morning. Now, I'd really like to introduce our industry partners, beginning with Pfizer, Dr. Rod McKenzie, who heads up Pharma Therapeutics Research and Development and is Pfizer's Group Vice President. Pfizer helped us break ground on this initiative. We've been at this for a very interesting few months and has been instrumental in helping us shape these agreements. So, Rod, can you say a few words about why Pfizer felt this was an important thing to do?
Yes, certainly. Thank you, Director Collins, Madam Secretary. Thank you for inviting us here to be a part of this initiative. It is a very exciting day for us. And we, as we've heard already, we invest very large amounts of our time and energy, and although much has been made of the amount of money we spend on our early research, what really impacts me personally on a day-to-day basis is the work and the hard work of all my colleagues in the laboratories. And as was pointed out, sometimes the initial idea we had doesn't play out when we get into the clinic. But we have these wonderful molecules that have all the properties they need to be useful clinical research tools. And too many times these compounds, they end up sitting on shelves or they end up in somebody's freezer. So what's exciting about this is that it gives us a chance to access on a grand scale. You called it crowdsourcing. I think that's very appropriate. The wonderful minds that we have out in the academic community. And we got kind of inspired in this because we had a relationship long-standing with Washington University in St. Louis doing this type of work. And what we realized was that the next step in all of this was to take it to a much, much broader collection of scientists and minds. And so the great hope that we have is that having invested in these molecules already, we may be able to connect with some great ideas and generate together some kind of data sets that may allow us to then make further investments and take these drugs all the way to the market. And I truly believe that these kind of public-private partnerships are exactly what we need as we create a new R&D ecosystem. It allows us to all do what we do best. NIH gets to invest in new knowledge. We get to think harder about the molecules, and we get to provide molecules to individual investigators. So we're very excited to be here, and thank you.
Thanks very much, Rod. Now let me ask Donald Frail, who is Vice President of AstraZeneca's Science New Opportunities Innovative Medicine Unit, to say a few words. AstraZeneca launched a similar arrangement with the UK Medical Research Council recently. So Don, can you say a bit about how that experience has helped AZ in this initiative?
Sure, I'd be glad to. I think as a little background, the MRC in the UK is the Medical Research Council akin to the NIH, I guess, in the US. And we had announced together in December a partnership to explore 22 different compounds that had been in development with UK investigators in partnership with the MRC. And I think there's a lot of learnings around that brought us into this initiative. And one in particular was a reconfirmation of our commitment to open innovation strategies in particular. And the early output that we've seen from the relationship has been quite strong. So in a short 8-week period for call for proposals, we've received over 100 different proposals covering 37 different institutions. So there with the crowdsourcing really working and actually on almost all of the compounds that we had offered. And it really struck us that this was indeed a model that we should commit to, and it helped us facilitate joining into the NIH initiative as well. I think another thing we learned out of that was that, frankly, the burden and the challenges on the company to identify the compounds and sign up to join. And we got a lot of comfort through the MRC initiative, a lot of learnings that exactly how to do that, that gave us, reinforced our commitment and gave us assurance to our senior leaders that this was the right thing to do. And then I think finally, in terms of relationships, frankly, simply relationships are important. And during this entire process of developing the NIH program, we've been able to share our experiences with the MRC to help them think about shaping how the program should be implemented. Some of the things might be a little bit different than the MRC initiative for good reason. And I think we had a great relationship with Kathy and the team in developing the different concepts and just sharing our experiences.
Great. Thanks very much, Don. To Don's left is Dr. Jan Lundberg, Executive Vice President of Science and Technology at Eli Lilly and President of Lilly Research Laboratories. Just 6 weeks ago, we had our first discussion with Lilly, and here they are today, a partner in this initiative. So Jan, say a few words on why Lilly felt it was important to be part of this initiative.
Thank you. I mean, in relation to that comment, for us it's very important to speed up the medicines to patients in need, and I really see this as an opportunity to do that. Lilly is an innovation company with a very long tradition then to see collaborations with academia result in life-saving medicines. I just want to remind everyone that 1923, even that early, Lilly worked with unique scientists at University of Toronto who then discovered insulin, and Lilly could make that available worldwide and for the dying patients of type 1 diabetes. In those days, the only treatment for diabetes was starvation, which I think we all realize is no treatment. We hope that by collaborating with the NIH, we can continue a very prosperous collaboration efforts that we have had. And I especially see this as a natural elongation then of our agreement that we recently made with the NIH, that Lilly is actually screening the NIH compounds in our models then in vitro in the cell tube testing then for potential disease applications. And now we are collaborating with our therapeutic agents with NIH and academia throughout the US for new potential applications. Why did it go so fast? I think we wanted to set an example that we have to do things faster to get the medicines to patients by not ending up in endless discussions about legal agreements. We are here to serve the patients, and remember, in the end, we are all patients.
Thank you, Jan. Now it's with great pleasure I'd like to introduce Michael Manganello and ask him to come to the podium. He is a living example of why this effort is so important. Michael learned in 1987 that he was infected with HIV and was one of the first to try the newly FDA-approved drug AZT. And while not a cure, this repositioned drug bought Michael time and hope. He's here today to share that story with us. Michael.
Thank you. Thank you, Dr. Collins. Good morning, everyone. I have to admit that this is a pretty exciting day for patients across this nation, and I'm honored to be standing here representing their voices. Thank you, Secretary Sebelius, for announcing this initiative today and for the support of this administration in recognizing that the medical research enterprise needs to think outside the box in order to bring cures and interventions to tens of millions of Americans across this country who are suffering and looking for leadership from the federal government. But I have to admit that the patients in this country owe a sincere debt of gratitude to Dr. Collins and his team for their vision. The National Center for Advancing Translational Sciences gives me and patients like me across this country something that you just can't put a price tag on, and that's hope. As Dr. Collins mentioned, I was diagnosed with the HIV virus, the virus that causes AIDS in the mid-'80s. I was 28 years old, and they gave me 18 months to live. As many of you may remember, at the beginning of the AIDS pandemic, there was not a single therapy or drug to stem the scourge of AIDS in this country. Yet in the 1960s, AZT was approved by the FDA to treat cancer and was soon shelved for lack of efficacy. 20 years later, a collaborative effort between the NIH and industry allowed the NIH to repurpose AZT because it proved effective against the immunodeficiency virus. AZT became the first drug approved to treat HIV/AIDS. All told, it took 25 months from the point when researchers learned AZT was active against HIV in the lab to FDA approval. It was one of the shortest development timelines in recent history. I started taking AZT in 1987, and while it wasn't the ultimate answer to my illness, I believe that it gave me the critical time that I needed for other interventions to be developed. By this time, I was enrolled in clinical trials at the NIH. In 1995, my participation in these trials resulted in me receiving the first approved protease inhibitor that saved my life. So thank you, NIH. However, a drug rescued from obscurity 25 years ago is the reason, and I have no doubt, that I can proudly make the statement that AIDS-related complications will most probably not be the cause of my death. It is very important to me that you understand, however, that I'm not just talking about HIV and AIDS drugs. I use this example because it's personal to me, and because it illustrates the outcomes that can be achieved through a model such as NCATS. NCATS and the initiative that we are announcing today is an important first step in identifying thousands of other drugs that have failed for one purpose but hold great promise for another use, like AZT did in 1986 for AIDS. I have spent my career working with patient advocates, caregivers, and scientists at the NIH, in academia, and in industry who have spent their lives searching for cures. There isn't one person in this country or in this room who hasn't been touched by ALS, Parkinson's, MS, Alzheimer's, spinal cord injury, cancer, diabetes, and thousands of other diseases and conditions. And for every American who is suffering, there is potentially an already abandoned drug waiting to be rediscovered. These are challenging times, both economically and politically. Everyone seems to have a hard time agreeing on anything. That I can be standing here at the podium supporting an initiative that brings the federal government and industry together for the good of the patients in this country is a remarkable thing to me. There is one truth that I can guarantee all of you. You and everyone you love and care about will at some point become a patient. Well, I guess there's a second truth that I can share with you also. Developing new therapies and cures is difficult. It will always be difficult, but if we all work together, patients, the federal government, and industry, we will get to our shared goal faster. I salute the 3 pharmaceutical companies here today for having the foresight to be pioneers in this initiative. Your commitment to curing disease will not be forgotten by the patients in this country. And to Dr. Collins and Kathy Hudson and the entire team at the NIH and the NCATS Center, thank you on behalf of so many of us for taking the first step. My plea, though, that this is truly the first step, with many, many more steps to come as we all go together down the road to cures. Thank you. Thank you.
Thank you very much, Michael. The Secretary will be able to stay for 2 questions. So we will begin with questions and ask the right ones related to this announcement. And so, but despite her schedule, she's been able to stay for the whole program and for 2 questions. So let's begin with questions directed toward the Secretary. And please, since this is being broadcast, please wait for the microphone. Otherwise, no one will hear you. Meredith? Oh, that's right. Very important. Please say your name and your affiliation, please. Meredith Wadman from Nature.
Testing. I am wondering, Meredith Wadman with Nature. Kathy said we hope other pharmaceutical companies will join in. Did you actually ask other pharmacos to participate? Did any decline, and why?
Well, actually, I'm going to let either Kathy or Francis answer that.
Well, we had actually a year ago a very interesting workshop at NIH on this topic, which was attended by multiple pharmaceutical companies, and it got a lot of interest, and it convinced us that there was a possibility of going forward here. These 3 companies were particularly interested in those discussions, and so we got to the point now of being able to announce a pilot, but we very much hope that other companies that have also been expressing interest all along will raise their hands and say, we want to be part of this as well.
Thanks, Meredith.
And I would just say I think the work that has gone on with Kathy and Francis and the team to develop the template really accelerates this. So I think people were waiting to see what this looked like in terms of how it could progress. And now that it's developed and launched, I do assume we'll have more interested partners.
Perhaps I could just say that, on behalf of my colleagues from the industry, I think we'd collectively be delighted if others of our colleagues across the industry would come and join the initiative, and you make it so much better.
Thank you. Question from John? I'll let you say who you are and where you're from, John.
John Reichart, Congressional Quarterly for Secretary Sebelius. What leverage would the government have if one of these compounds pans out to negotiate favorable pricing?
To negotiate favorable pricing in the marketplace?
That was an issue with AZT and the AIDS drugs.
Right. At this point, I think the pricing issues clearly are not in the either NIH or FDA. FDA is in fact prohibited from looking at pricing as they move a drug to marketplace. We would have some leverage at the end of the day in the Medicare program, certainly the Medicaid program, which we run, to do various negotiations. But in terms of launching the drug in the marketplace, we don't set prices, we don't control prices. That's really up to the industry partners.
Yeah, let me say that if there are intellectual properties that are invented by the partner, the academic or the biotech company, whoever the applicant is who gets access to the compound, then presumably, in the usual way, there would be some royalty sharing as a consequence of that, so that the partners would also have some potential benefit for a compound that's successfully marketed. But again, there is no intention as part of this program to intervene in terms of pricing. That is something which NIH, 15 years ago, was asked to get a bit more engaged in, and it became quite clear that that was a non-starter in terms of productive relationships with the private sector.
All right. Thank you very much, Madam Secretary, for joining us this morning. Appreciate it.
I will. Thank you.
Thank you. And please, your name and affiliation.
Emily Walker with MedPage Today. I have a question for the drug companies. Colin said that it takes about $2 billion in 14 years to move a drug from the lab to the clinic. What's kind of the investment of these compounds? How many years did it take, and what kind of money did you spend? My second question is there any concern of, what are the competition concerns between the companies, that another drug company might take this data on this compound and do something on its own with it?
Well, I can start with the first question, and then I'll let my colleagues deal with the second question if they agree, because I'll forget it. These compounds are, I would say, somewhere about halfway along the development program. Probably somewhere between 5 and 7 years in, and certainly tens of millions of dollars in. You've got to remember that the $2 billion that gets quoted is not the cost of an individual medicine making it to the end. It's actually the cost of that molecule plus all the ones that don't make it rolled up. So, it's certainly tens of millions. It's certainly 5 to 7 years, something like that. And so the good news is that when we find ideas, we're that much further along, and the compounds already have demonstrated enough quality to be in human beings.
Thank you. Yes. Oh, another comment?
There was a second part of the question.
Second part of the question, sorry.
Yeah. Well, I was going to also say on the investment part, we still significantly continue to invest in these compounds in the fact that we have to maintain drug supply and provide it to the investigators. We have to maintain patent costs and things like that. So we are still committed to these compounds and trying to see them through to patients in other ways. In terms of competition, actually, we're very open to publication, and it's part of the agreements where we will be encouraging the investigators to publish their work, and more open access that will actually benefit us all, in particular the patients. If there's a failed clinical study, that will in itself be important, and if we can get it published, it'll learn from that and move on to other more productive hypotheses.
Okay, thank you. Trying to get you in order. I think Alex, you had your hand up, Alex, and then next to him, then David Brown. Okay.
Hi, this is Anna Yukonano from Reuters. I was wondering if you could just give an example of the 20 compounds that you're first starting off with. And also, you mentioned 4,500 diseases that we've discovered the causes. What are some examples of those that you're looking particularly to focus on? Thank you.
So we're not releasing information about the specific compounds today. So there are 24 compounds that have been made available by the companies. We put out a request for information today about the program and for comments on the model template agreements. When the request for applications is released in about a month, it will link to summary information about all of the compounds. We hope that the 24 grows between now and the time that we put out that request for application, but we're not discussing specific information about those compounds today.
Don, you had a follow-up?
I was just going to follow up that as part of the AstraZeneca MRC relationship that was announced in December, the compound list for that one is posted on an open website for anybody to see. So if you want examples of what may be included, those are there to view and give you a very good flavor, I think, of types of things that might be included.
Francis.
And it is a broad range of compounds that will have, I think, great interest in the community. In terms of your question about diseases, those 4,500 diseases, many of them are rare conditions. Many of them are genetic conditions where one knows now what is the molecular cause by the process of a genomic analysis. While they are rare, if they're in your family or in yourself, they're extremely important for new efforts to be mounted. And the fact that we have discovered at the DNA level the exact cause of those diseases means that we have a very good idea about what kind of drug one would want to see and what target it would need to hit. So there's a particularly good opportunity there. Some of those are neurologic conditions. Some of them affect other parts of the body. Almost any part of the body can be affected by one of these rare genetic conditions, and they are urgently in need of attention.
Alex, and then David, and Jocelyn.
Kind of a follow-on to that question, Alex Wayne from Bloomberg. Are there any particular diseases that you're hoping will be, I guess, these compounds you picked will be effective in against some particular disease or some particular pathogen?
Let me just say that I think many of us are hopeful that approach to CNS diseases will be particularly advantaged by this set of initiatives. Those are very difficult conditions to tackle, and many pharmaceutical companies have actually found it necessary to step back in some of their R&D efforts. Not all, but some have done so because of the long lead times and the uncertainty of the field because of things like the blood-brain barrier. And yet we're learning a lot about diseases of the brain. So this would certainly be an area where I would hope there would be a lot of activity.
I mean, maybe just add to that and be even more specific, I guess, because I agree with Francis that the neurological disease is one of the great health crises that we face, certainly across the globe, and we need to do better there. And so, if I had a wish, that would be that we would find some insight there that would lead to breakthroughs in Alzheimer's disease, but also diabetes and cardiovascular disease, still the biggest killer and an epidemic proportions in this country. So, there are many, many areas that we'd love to work in, but I just highlight two particular ones of some passion for me.
Okay. Anyone else on the panel? Okay. David.
Yeah, David Brown from the Washington Post. Can any of you mention some other examples other than AZT and raloxifene? And then a sort of related question is, all of these compounds are presumably failures in some way. Do they all have sort of clear, likely applications in which, their activity and the pharmacology, it's all there in plain view, it's just not part of the pharmaceutical company's portfolio? Or in fact, are you talking about making available a very small number of compounds which will then have to be screened? Because we know that when you screen chemical compounds, you have to screen tens of thousands before you get a hit. And if we're talking about 24 compounds, it would seem to me it would hardly be worth the effort unless it's really pretty clear what these are, what the usefulness of these compounds are.
Yeah.
Yeah, let me just get back to the stage of these compounds and the type of quality that they represent. These agents have been very thoroughly then investigated and optimized and safe to be taken into human testing. initial data from human volunteers will be available, and probably also testing in some patients where the outcome, though, wasn't enough to be sufficient for progressing into phase 3. So I think we see here, it's actually, if you would compare to a relay, you are actually starting as person number 3 out of 4. In a way, so much work has been done here, so it's relatively easy to test new indications for these compounds, and they are clearly selective, safe, and you also have the dosing schedule established, and in many cases you have a biomarker so you can measure if you have the right type of effect on this particular mechanism. Remember, though, that even if the human genome was cloned and there are thousands of different potential disease targets, not all of them, can be applied to certain diseases. So I think it's highly relevant to search for new indications for selective agents. And the key thing here is to have agents that are not probes. They're good enough to give conclusive data, which of course is very important for the patients that are waiting.
And Francis?
And these are compounds where you know, for the most part, the specific molecular target. This is not just a random compound you picked out of a library of chemical shapes. These are compounds where you have a lot of molecular information. So the idea is to do this matchmaking. Oh, on this list, we have these compounds that hit those targets. On this list of 4,500 diseases are places where we know about the molecular pathway that is involved. Could we put that together? This is not a random screen. This is very much an intentional, designed connection between the drug and its potential application.
Okay,
and you asked also about other examples of where this has happened. There's a long list, maybe a rather famous one, thalidomide, the drug that was developed for morning sickness, obviously a disaster in that situation because of its potential to cause birth defects in pregnant women. But thalidomide has now been repurposed as a very effective drug against multiple myeloma. Now, who would have thought that those things would somehow be connected? And yet, there we are. Recent publication about Alzheimer's disease, this drug bexarotene, which was developed for T-cell lymphoma, and in the mouse model, in this dramatic paper in Science about a month ago, shows the ability in a very short period of time to result in reductions in amyloid in the brain and increased mouse performance in the best model we have. Just a few, there's dozens of these, but they've all been sort of serendipitous. Somebody had an idea. The idea here is let's not depend on serendipity. Let's do this in a more systematic, comprehensive way by going from individual ideas to the crowdsource model.
Thank you. Jocelyn Kaiser from Science.
Yeah, so just to follow on to that question before, so you described sort of the criteria for including a compound in this set of 24, but I'm wondering, what's the universe of compounds that have reached that point, and how did you pick this set of 24?
I think, clearly what we wanted to do was pick drugs that had very, very good qualities so that researchers could immediately, without further work, go into the clinic. Because the idea here is speed. Once we have the idea and we have the funded proposal, we want to go straight in, and we don't want to have to do further research on the molecule. So that, as Jan and others have pointed out, that constricts the universe pretty dramatically. It's not the start of the screening funnel where we have thousands of compounds. We're right down to compounds that are in phase 1 or phase 2 human testing. So now you're into the world of dozens of molecules. And, I think in every pharmaceutical company, there's a limited set of those that, on the one hand, have not achieved everything we hoped in their first indication, but still have the right kind of qualities for further clinical study. As you know, many compounds, or too many compounds, still reach the clinic, and we find out something about safety that we don't like. And so they get stopped. We don't put any of those compounds into this initiative. These are all compounds where we're very confident we can do safely clinical work in humans, and that's why it's that kind of number.
Don.
Yeah, I was going to be provocative too. One of the criteria that the NIH had was that they were discontinued, not currently active in the development portfolio, which was actually not a criteria of our MRC relationship, and I tried to be provocative with Kathy and NIH to consider actually that maybe it's not necessarily restricted to discontinued compounds only. certainly there's been quite a bit of work at the NCI around compounds that are still alive and active in certain development portfolios. So I think maybe that's a next frontier. Certainly not part of this program, but something for further discussion.
This is a pilot.
Before we go to follow-up questions, that's fine. Yes.
Donna Young from Scrip. I wanted Kathy just to talk a little bit about that whole negotiation process, how that went, how many lawyers were involved. If you could maybe kind of explain, how smoothly or how rough that went, and how long of period that actually took for you to do that. And then my follow-up to that would be, at what point then, if you've got the researcher working on this, at what point can the drug company jump in and take that further? I'm assuming that's all part of that template and included in there.
Right. So let me say a word about the experience of negotiating with 3 companies at the same time. So none of the companies knew who the others were, or at least we don't think they did. And we had an introduction about a week ago on a phone call that was a little bit like the matchmaking. Everybody got introduced. It has been a wonderful experience from my perspective because I've been able to learn so much about what goes into the drug development process. Pfizer was the pioneer here and started negotiating with us some time ago, and folks are sort of in the order in which they came to us and started to negotiate. I think this has really been a profound source of a new way of working together. I don't think NIH had this scale of collaboration with the private sector in the past, and assuming that other companies join, this will be really, really profound. In terms of, what was your second question was?
At what point then can we...
Oh, going back. Right. So there are 3 sets of agreements that sort of govern these relationships. There's MOU between us and the companies. There are collaborative research agreements templates between the companies and the grantees, and then there are the grants, which govern the relationship between us and the grantees. So this is a complex set of 3 two-way agreements, and they all hopefully will work in great harmony together. So in the collaboration research agreement, it lays out the terms for intellectual property that's developed by the researcher, and the ability of that intellectual property to be licensed back to the initial company, but also providing for the opportunity, if the company does not want to pursue it, for a third party to be identified that could carry that forward. So in our negotiations, we always felt like we were there at the table negotiating on behalf of the American people and patients, and we always wanted to make sure that at the end of the day, if there's something promising, we want it to be pursued, and I think these agreements do that.
Okay. Meredith, you had your hand up. Anyone else before? Oh, Tom Burton hasn't had a chance. Then Alex and Meredith are okay as well. Okay.
That pillar really helps a lot, doesn't it?
Hi, Tom Burton with the Wall Street Journal. You've been circling around this issue, but I'm wondering if is it possible to be at all more specific? If one of these drugs is actually successful, how do the profits get divided among the researcher, the company, and the NIH, the public?
So Kathy, do you want to talk about what the current agreement says, which is that that's something that gets negotiated between the company and the investigator at the time the project is initiated?
Right, there's already existing law and policy that governs a lot of these relationships. So if additional intellectual property is developed by the researcher, then it will be the researcher and his or her institution that own that intellectual property and can license it back. And those terms are negotiable, and they're negotiable in terms of the value that has been added by that additional piece of research. NIH has always made an enormous contribution to the development of new medicines. Traditionally, our investment has been very, very early in the pipeline. In this case, our investment is happening in a different place in the pipeline. But I think that the mission for the NIH to do what we can in an open access environment to speed the development of new drugs, that's what this program's about, and it's not distinctly different from other NIH research initiatives.
Okay. Kathy, and then Meredith, and then Alex, and then we'll wrap up.
Kathy Dombrowski from the Pink Sheet. Could you give us a little bit more information about how are you going to select the researchers and which compounds they're going to look at?
So can I take that one?
Yeah, take it.
So when we put out the request for applications, that will be broadly accessible to the entire research community, both academic-based, nonprofit, research institutions, biotechnology companies, the whole horizon. Individual investigators will have access to the summary information about the compounds that is not unlike the information that AstraZeneca has with MRC. And so you can sort of get an example, a flavor of what that information looks like. So it'll include mechanism of action, dose, and so forth. The investigators who have specific disease interests and expertise might see something on that list and decide that they want to try to get access to that compound and the funds. They will submit a summary application that will be reviewed in the traditional NIH peer review manner. Those who are successful will be invited to submit a full application, and in that full application they will have to demonstrate that they have access to the compound. The way they will demonstrate that they have access to the compound is that they will have signed the collaborative research agreement that has already been pre-negotiated and which you can access on the web today. So the great thing about that step is that right now those negotiations, as Francis mentioned, often can exhaust time, resources, and people's patience. So hopefully these pre-negotiated templates will speed that process up. Those applications will come back in, and those that go through another round of peer review, and we will award $20 million in next year, probably in about April or May.
All right, thank you. So Meredith, and then Alex, you'll get the last question.
Meredith Wadman with Nature. A quick question, then a more complicated one. Are these like $21 million one-year grants? Are they more like standard RO1s, or they extend over 5 years? That's the first question. The second is, AZT was already approved, and so I'm kind of confused by, you seem to have strictly limited this initiative to pre-approval phase 1, phase 2 candidates. And are there simply explained IP-related reasons why you chose this group of compounds rather than post-approval compounds? And is that an important question? I'm not even sure. I'm trying to figure that out.
So on the first question, the mechanism, the grant mechanism that we're going to be using is a cooperative agreement. And so we will ask investigators to lay out very clear milestones that they will need to meet. And so in the first phase, presumably in the first year, they will validate this compound in their preclinical model for their indication, and then move quickly into human studies in year 2 and possibly 3. So this is a cooperative agreement mechanism where after the first year of milestones haven't been hit, there won't be additional funding. So this is a non-traditional mode. We've used it some at NIH, but it's going to be particularly valuable here to make sure that our resources are well spent. We have $20 million in the first year. These will be 2 to 3 year awards, so there'll be additional dollars in the out years, presumably escalating a little bit because the clinical part starts later. And then in terms of why did we pick compounds that are no longer being developed by the companies and not approved for another indication. So we certainly would like to explore that. In our discussions with companies, there are more complexities to supporting research on a compound that already has been approved for one indication for many reasons. So the legal complexities here were already substantial. I think they would have been overwhelming for a pilot to try to move into repurposed compounds. So we'll certainly continue to explore that and hope to grow into that area. But we
another thing to say there is that this is a group of compounds that are not easy to get access to historically because they sit within companies and the data is retained within the company, and so this is the first time that kind of compound's available, which is another reason to go for this group.
Okay. Francis, did you? Okay.
Do we have Alex?
But Alex, and then, yes. Oh, go ahead. Okay.
Thank you. Jessica Zygmunt with Modern Healthcare Magazine. Just a clarification on the $20 million over 3 years. Is that just for these three companies, or you said, you know, you're welcoming other industry leaders. Would that be open to them as well? Thanks.
So I'm sorry I wasn't clear. So there will be $20 million in FY13 and an additional approximate 20 in the following fiscal year and the following fiscal year. So this is just for the first year. These are dollars from the National Center for Advancing Translational Sciences. We are hopeful that that other institutes and centers at the NIH will also be interested in supporting projects. So imagine that somebody submits an application that's related to cardiac indication. Then it might be that the National Heart, Lung, and Blood Institute would be interested in co-funding that application or picking it up entirely. So we have this $20 million, which is may be catalytic for greater investments across the NIH.
And again, be clear that the awards then go to the investigator who's proposed the new use for the compound. The companies are contributing the compound and the data, but they're not receiving dollars from NIH.
Right. Okay, thank you. Alex?
Alex Wayne from Bloomberg again. I just wanted to sum up my understanding of the financial relationships and then ask a question. So it sounds as if NIH will provide some grant money to the researcher. The company gets sort of a right of first refusal if this leads to some discovery of efficacy. And then the company and the researcher work out sort of the profit sharing or whatever. But as best I can understand, there's nothing that comes back to taxpayers from these deals except for the societal benefits of the medicines.
Yeah.
Yeah, just a comment, you know, I think when you look at the benefits of new medicines, you have to have a holistic view on the society. I mean, if people can get back to work, they generate job taxes, and also pharma companies or biotech companies, if they have sales, etc., it will be taxes back to the US system. So in a way, you could even say that the taxes from companies in the US are funding the NIH. So you have to look more holistically, I think, when you talk about where does the potential profits go. But in the end, I think it's for the benefit of the American healthcare system.
Well, now, it would depend in terms...
Funny you ask that.
Yeah, it depends, of course, who the investigator is. If the investigator happens to be an intramural NIH scientist and intellectual property results, then there would be a royalty stream, presumably, that would flow back to NIH, which would then fund more research. If the investigator is at an academic institution, because of Bayh-Dole, the intellectual property would actually reside with the institution. They also would take their proportion of any royalties and use it, presumably, to encourage more research. So in that regard, there is a payback to the taxpayer. Keep in mind, while we're talking about $20 million, and that's a big investment for NCATS just getting started, we're only 4 months old now, consider that each of these compounds has already had invested in it tens of millions of dollars and years of work. And what we're trying to do here is, with a modest investment, bring that previous effort and all that it entailed to a new life. So one shouldn't overstate, I think, the contributions that we're making through this program to what has already been done to bring these products to the point where we could contemplate a program of this sort.
Okay. Thank you very much. We'll also be available if you have other questions. Dr. Collins, if you just want to...
Well, I can tell by the wonderful questions we're getting that this is an interesting program for all of you, and it certainly has been for us. For myself, as the director of the NIH, one of the driving forces that all of us think about every day is how can we speed up the opportunity to bring new treatments to patients whose hopes depend upon that. And especially now when resources are quite tightly constrained for the public sector, for the private sector, coming up with creative ideas about how we can do this more efficiently and more cost effectively is something we think about every day. I am just delighted that we are here today on May the 3rd after a year of hard work with the companies and with really dedicated folks on the NIH staff, and Kathy Hudson leading that, but I also want to mention Linda Brady, who's standing over there, who's an enormous contributor to this whole enterprise. And I am looking forward to seeing how this plays out. So watch in a month when the RFA comes out, but even now, watch today for the request for information, for an opportunity to look at all these templates that have been put together, which is itself a major contribution. And let's see what we can do. I'm hoping that out of this will come some real advances that will help people who are currently waiting for us to come up with answers. Thank you.
Thank you very much, and thanks again, everyone, for joining us. Thank you for the panel.